PhenolRed test, determining fermentative properties of different bacteria. The PhenolRed test is a simple broth inside of a test tube. Placed upside down in the broth is a smaller second test tube called a Durham tube. During fermentation, CO2 gas is often given off. If CO2 production is occurring during fermentation by a bacteria in a PhenolRed broth, gas or CO2 will be trapped in the Durham tube and will be made visible. For this experiment, you will be inoculating two different PhenolRed broths for each bacteria, a glucose and a lactose. Glucose is a monosaccharide, lactose is a disaccharide. Prior to inoculation, since both broths will look exactly the same, you need to label the test tubes as to which one is glucose and which one is lactose. Bacteria capable of glucose fermentation sometimes are also capable of lactose fermentation. However, an organism capable of lactose fermentation is always capable of glucose fermentation, since lactose is a disaccharide and glucose is a monosaccharide. When lactose is fermented, an enzyme known as beta-galactosidase will break the disaccharide into two individual molecules, galactose and glucose. Galactose will be converted into glucose, and glucose itself will enter into glycolysis for fermentation. The inoculation of the PhenolRed broth is a simple broth inoculation. Use aseptic technique, obtain your bacteria, and carefully inoculate each broth separately. The loop should be flamed in between each inoculation, even if you are using the same bacteria. During inoculation, it is also important to make sure that the entire loop is flamed and sterile. It is also important to allow the loop to cool sufficiently between inoculations. Inoculation of a PhenolRed broth with dead bacteria will actually have no results at all. You also do not want to over-inoculate the tube. Over-inoculation can cause the sugars to be used up too quickly, resulting in protein catabolism, or a property of some bacteria known as reversion. This can make results or interpreting results very confusing and difficult. When you have completed the inoculation, be sure to sterilize your loop. It is not necessary to remove all of the bacteria off of your loop during inoculation. You simply need a few cells to fall off of the loop into the broth in order for the population to grow. Here is fermentation occurring in a glucose broth. On the left is gas production, shown by the bubble in the Durham tube. On the right is no gas production. Notice that fermentation did occur in both of the broths, causing acidic conditions which makes PhenolRed, the pH indicator, turn yellow. This broth is a non-fermenting bacteria. Instead of fermentation of sugars, the bacteria used protein catabolism, or protein breakdown. This results in alkaline conditions. Alkaline conditions cause PhenolRed, the pH indicator in the broth, to turn a bright pink. Gas production does not occur during protein catabolism, so there should never be a bubble in the Durham tube in a protein breakdown PhenolRed broth result. Now shown are the possible reactions that can occur in a PhenolRed broth test. On the left is a control, or uninoculated broth. The first tube is called alkaline, or K/negative. No fermentation has occurred, and instead proteins were broken down, causing alkaline conditions. This causes PhenolRed to turn a bright pink. The third tube is A/positive, or A/G. Fermentation has occurred, giving off acidic conditions. In acidic conditions, PhenolRed will turn bright yellow. Also, for this tube, gas production occurred during fermentation. CO2 was trapped in the Durham tube, forming a bubble which is visible. And the last tube is A/negative. Fermentation did occur, giving off acidic conditions, causing PhenolRed to turn bright yellow. However, the Durham tube still has fluid in it. There is no bubble present in the Durham tube, indicating that no gas production or CO2 production occurred. The PhenolRedBroth test is a very useful test for determining what sugars a bacteria can or cannot ferment. It is used in identification of gram-negative enterics and is also useful for some gram-positive organisms. It is often one of the first biochemical tests used in the identification of bacteria.